Good evening, good afternoon, everyone. Today we have here Professor Vincenzo Di Marzo. He's going to, we will have a talk about the endocannabinoid system. Professor, you can start your talk whenever you're ready. I'll be here if you need anything.
Thank you very much, Beatrice. Thank you for to the organizers for inviting me to this online presentation. I'm sorry I cannot be there in person, but maybe one day when the situation will be better all over the world, we will start traveling again, and it will be easy again to travel. So today I've been asked to give you a general overview of the complex pharmacology of cannabinoids, which are the most peculiar bioactive natural products found in the cannabis flowers, the flowers of the cannabis plant. And from there, I will move on to describe briefly, because you probably know already all about it, the endocannabinoid system, and more in detail, the endocannabinoidome and some new concepts regarding these two complex signaling systems that were discovered starting from the cannabinoids. So just the disclosures to just let you know that I've received grants and consulted for GW Pharmaceuticals. And what I will basically tell you today is how we went from the discovery of the cannabinoids to the discovery of the endocannabinoid system, how there is a whole lot of cannabinoids beyond THC, and this led to expand also the endocannabinoid system to the endocannabinoidome, and how most phytocannabinoids do not target the endocannabinoid system, but instead interact with the endocannabinoidome. How most phytocannabinoids are multi-target compounds, and therefore understanding their mechanism of action means that we need to appreciate the value of polypharmacology that can often be more therapeutically useful than the magic bullets represented by synthetic drugs. And of course, I will try to just very briefly show you some unifying theories on the mechanism of action of phytocannabinoid pharmacology, interactions with the gut microbiota, which is what I'm working right now, and the future developments. So as you all know, the discovery of the endocannabinoid system followed more or less in the same line of thought as the discovery of the endorphin system. The endorphins were discovered first when it was found that some endogenous peptides could mimic the action of morphine, and the discovery of these endorphins led to the discovery of the opiate receptors, first of all the mu receptor for morphine. Similarly, the discovery of THC as the major psychotropic and psychoactive principle of the flowers of the cannabis plant, in this case, first led to the discovery of the receptors for THC, delta-9 tetrahydrocannabinol, which are two G protein-coupled receptors, the CB1 and CB2 receptors that you see schematized here in a bidimensional way, quite different from each other. They only share about 40% homology. And of course, the fact that these receptors exist meant that some endogenous ligands for these receptors should also exist, and these were discovered at the beginning of the 90s by the group of Rafi Meshulam. And as you can see here, these are anandamide and 2-arachidonikglycerol, which are derived from arachidonic acid. Clearly, the endocannabinoid system, therefore, besides finding the two endocannabinoids, which, as you can see, share some chemical similarities with THC, for example, this N-pentyl moiety or the hydroxy group, they are quite different. They are fatty acid derivatives, the ester with glycerol and the amide with the ethanolamine of arachidonic acid, but they share some of the pharmacophore of THC, which means that they can interact with the same receptors. But of course, the way of these compounds being produced is completely different from what happened for THC in the plant. Anandamide and 2-arachidonikglycerol, or 2-AG, are, as you can see here, produced usually on demand, which means that their concentration in a non-simulated cell are usually very low, and it increases when there is an increase in intracellular calcium concentration. And this means that normally the cell is stimulated with some kind of stimuli, which this elevation of calcium stimulates the enzymes that produce anandamide and 2-AG from phospholipid or lipid biosynthetic precursors, which in turn comes from the remodeling of phospholipids. And once these two endocannabinoids are produced on demand, they activate the CB1 and CB2 receptors only locally to play a pro-homeostatic function. And once they have played this function, they are taken up by the cell and inactivated by intracellular hydrolytic enzymes, the fatty acid amide hydrolase and the monoacylglycerol lipase, two degradation products that are inactive on the two cannabinoid receptors. And in this way, the signal brought by the endocannabinoids by acting on CB1 and CB2 receptors is terminated, like all chemical signals. I mentioned that this system of on-demand production of the endocannabinoids is a pro-homeostatic system. What does it mean? It means that the activation of the system, the activation of cannabinoid receptors in the cells, in the tissues, in the organs, usually comes after a transient or a permanent perturbation of the homeostasis, and has the task of bringing the cell or the tissue or the organ back to the situation that was before this perturbation. In general, we can say that the activation of the endocannabinoid system by endocannabinoids, by anandamide and 2-AG, unlike what happens when you assume THC systemically, happens in a manner that is very selective, both in space and time, to produce a series of events that help, which are shown here, and these functions help the tissue, the organ, or the organism to go back to the steady state and to recover from stress, for example. As you can see, both cellular stress and psychological stress can activate on demand the endocannabinoid system, and through the activation, namely of CB1 or CB2 receptors, this brings back the organism to a situation of equilibrium, and therefore, in some way, we can say that it fights against the perturbation of the homeostasis. However, there are also some pathological conditions in which the endocannabinoid system is activated in a manner that is not selective in space or time. And this happens, for example, in some chronic pathological conditions such as obesity, metabolic disorders, some neuroinflammatory disorders, inflammation, chronic inflammation, where the over or misplaced activation of the endocannabinoid system and CB1 and CB2 receptors can contribute to the progress of disease and symptoms rather than fight the disease. And this is why the endocannabinoid system was considered as a complicated but also quite challenging and exciting target for the development of new drugs, because you can imagine that both agents that activate CB1 and CB2 receptors, mimicking the actions of the endocannabinoids, either directly or indirectly through the inhibition of the degradation of the endocannabinoids, but also synthetic agents that antagonize the CB1 receptors could be used depending on the situation, depending on the pathology, depending on the phase of the disease, as therapeutic agents. Indeed, we know that there are still on the market some endocannabinoid system-based therapies, such as, first of all, THC, of course, whose use is still used in the US and in many countries against nausea or cachexia in oncological conditions or ADS patients. Of course, we must remember that some of the side effects of THC, which are the same as some preparations of cannabis rich in THC, like marijuana or hashish, produce psychotropic and euphoric actions that are not always desired in these patients. So the use of THC in the clinic has been very much limited by its central side effects that are the basis for the recreational use of marijuana, but are not really desired in pathological conditions. But there have been also, as I mentioned, cases in which blocking or antagonizing the CB1 receptors could be therapeutically useful, and this antagonist, rimonabant, has been on the market for almost 3 years. It was very efficacious against obesity and type 2 diabetes, because in these conditions, the endocannabinoid system is overactive at CB1 receptors. So antagonizing the actions of the endocannabinoids at CB1 receptors was efficacious at reducing obesity, body weight gain, and all the metabolic consequences of obesity, such as steatosis, insulin resistance, glucose intolerance. Again, because of the presence of CB1 receptors, which are the most abundant G protein coupled receptors in the brain, in the mammalian brain, also the use of these antagonists is very limited, and rimonabant had to be withdrawn from the market because it was producing depression and anxiety in these patients. Why depression and anxiety? Because, as I mentioned before, the endocannabinoid system, particularly in the brain and particularly via CB1 receptors, is important to cope against stress. So if you use a blocker that goes into the brain, yes, you block some metabolic effects, but you also reduce the capability of human beings and animals to cope with stress, and therefore you produce pro-depressing and pro-anxiety actions. This is why CB2 receptors and also inhibitors of the degradation of the endocannabinoids are now being investigated, because they are supposed to have less side effects, central effects on mood or on the brain. And they are still under development. Some of them have been tested quite disappointingly in the clinic. So we still have no example so far of CB2 receptor agonists and phi inhibitors that have gone to the clinic. This is why perhaps many researchers working on the endocannabinoid system have turned back again to plant cannabinoids, the phytocannabinoids, and have realized that THC, which is a difficult compound to work with when you want to develop a drug, a therapeutic drug, beyond THC, there are many other plant cannabinoids that until the beginning of this century had been quite neglected. Everybody knows cannabidiol these days, but I can tell you that at the turn of the century, very few people were working on cannabidiol, and also some other like cannabigerol, cannabichromine, and the delta-9 tetrahydrocannabivarin, which despite very chemical similarity with THC, is not really able to activate efficiently the CB1 receptors. So we are working on many other people started working on these compounds, and there is many others beyond these four, like the propyl and the methyl analogs of these molecules, the sesquiterpene analogs of cannabigerol. It is now estimated that there is over 110 cannabinoids molecules with this kind of structure, which is mixed with, between polyphenol and an acetate-derived biosynthetic compounds, and people have started investigating their pharmacological activity, and this is what I'm going to talk about today mostly. So as I said, there is more than 100 such compounds. We only know the pharmacology of a handful of these molecules, so there is a lot of work to be done. Why is it important to understand the molecular mode of action of these phytocannabinoids, and what do they have to do with the endocannabinoid system? Well, most of them very little, except tetrahydrocannabivarin, which is a neutral CB1 antagonist and a weak CB2 agonist, and all the other non-psychoactive, non-psychotropic, non-euphoric cannabinoids like cannabigerol have only, or cannabidiol, have only little activity at cannabinoid receptors. There is now, however, evidence that cannabidiol may act as a negative allosteric modulator of CB1 receptors. This means that it binds not on the orthosteric site where THC and other agonists bind, but binds on an allosteric site and reduces the activity of THC. This is interesting. Of course, and this is however, there are many other ways through which cannabidiol can in some way tone down the psychotropic activity of THC, and this is why those cannabis varieties where the amount of CBD is comparable to the amount of THC do not produce some of the same euphoric or even negative effects on like unveiling the psychotic activity of THC on certain predisposed subjects. So there is evidence, however, that some of these compounds can interact with the endocannabinoid system. For example, cannabidiol can inhibit anandamide hydrolysis by Fa, and these other compounds can inhibit the biosynthesis of the other endocannabinoid 2-AG, and most of them can weakly inhibit the cellular reuptake of endocannabinoids from cells. I mentioned this at the beginning. This is a process which has not been molecularly characterized yet, but it's definitely there. Endocannabinoids need to be taken up in a non-passive way by the cell membrane in order to be degraded, and these phytocannabinoids weakly inhibit this process. But as you can see here, most of these actions are really not very strong actions, and therefore people have started looking for different targets for these non-psychotropic cannabinoids, targets that are outside the endocannabinoid system. And this is also one of the reasons why the endocannabinoid system was recently expanded to the endocannabinoidome. The other reason being that anandamide and 2-AG, the two endocannabinoids acting on CB1 and CB2 receptors, are accompanied in tissues by many other chemical congeners. Anandamide is accompanied by other N-acyl ethanolamines, and 2-AG is accompanied by other 2-acyl glycerols, and there is also fatty acid amides between fatty acids and amino acids. You can imagine how many of these can be produced by cells, considering that there is several long-chain fatty acids and at least 20 amino acids. So we can have up to 200 lipoamino acids or acylated amino acids. We also found that some fatty acids can make amides with the neurotransmitters such as serotonins, dopamine. And you can see here that all these endocannabinoid-like molecules in most cases do not interact with CB1 and CB2, but interact with other receptors. And some of these receptors are G protein coupled receptors like CB1 and CB2. Some others are channels like the vanilloid TRPV1 channels. Some others are nuclear receptors such as the PPAR alpha and PPAR gamma. And it's important to know this because, we can modulate the levels of these compounds with the diet. You can imagine that each of these compounds would be more or less abundant in our body, in our membrane phospholipids, depending on how much oleic acid or linoleic acid or docosahexaenoic acid or palmitic acid we consume in our diet. And we recently discovered this is really the case. Most of these compounds are biosynthesized or degraded by biochemical pathways similar to anandamide and 2-AG. This is true for the N-acyl ethanolamines and 2-monoacyl glycerols, but you can see here that there is a redundancy of this pathway, so it's very complicated. The N-acyl amino acids and the N-acyl neurotransmitters instead are biosynthesized via different pathways. And the degradation can happen via hydrolytic enzymes, but also for those members of the endocannabinoidome or endocannabinoid-like mediators that contain at least two double bonds, like linoleic acid, arachidonic acid, and many others. There is the possibility of these compounds being also oxidized by the so-called arachidonic acid cascade enzymes, such as the cyclooxygenase 2, the lipoxygenase, and cytochrome P450. And what's interesting is that in most cases, the oxidation products of these endocannabinoids and endocannabinoid-like molecules are not inactive at CB1, CB2 receptors, or are inactive but are active at other receptors, for example, as shown here for the so-called COX-2-derived prostamides and prostaglandin glycerol esters. So the family of the endocannabinoid-like mediators includes, I would say, at least 200 endocannabinoid-like molecules, but also at least 15, 20 enzymes and at least 15 receptors. And it's important to see how this endocannabinoidome can become the target of the phytocannabinoidome. THC, as I said before, is accompanied by other phytocannabinoids in the cannabis plants or their acid derivatives. And here I only showed 16 most studied cannabinoids in the cannabis plants. And if you depict the endocannabinoidome here, this is a very out of date picture of the endocannabinoidome, where only a few receptors and a few enzymes are shown. You can see that, in fact, even though most of these compounds here are not able to bind to CB1 or CB2 receptors, they can interact with the enzymes or of the endocannabinoidome, or with other non-cannabinoid receptors of the endocannabinoidome. In particular, the TRP channels. This is a family of cation non-selective channels that are usually activated by physical stimuli such as temperature, high temperature, low temperature. They have a feature that are activated and immediately desensitized. So sometimes you cannot really distinguish what is the effect of their activation from the effect of their inactivation, because the inactivation through desensitization immediately follows the activation of these channels. And as I said, these are completely different from the G protein coupled receptors. These are 6 transmembrane domain ion channels. There is a large family of them, and 6 of these channels, TRPV1, 2, 3, 4, TRPM8, TRPV1, apart from being activated by either low or high temperature, this is their main function in sensory neurons, when they are present in other cell types, they can play other functions. And another interesting point is that many of these channels are activated by natural products. You see here TRPV1 activated by capsaicin, which is the hot pungent principle of hot chili peppers, and TRPM8 being activated by menthol, which mimics the activation of cold. And so several years ago, we wondered if the plant cannabinoids could also activate these channels. And indeed, this was the case. And here you see a summary of all the actions that have been discovered so far, with cannabidiol, tetrahydrocannabivarin, cannabigerol. You see many of them activate and desensitize TRPV1, the capsaicin receptors, or antagonize TRPM8, the menthol receptors. Some of them activate TRPV2, others much more weakly activate TRPV3 and TRPV4, and they all activate, except for the fatty acid phytocannabinoids, they all activate TRPV1 channels, which is also known as the mustard receptor. As you know, these TRP channels recently, last year, in fact, got Nobel Prize for their discovery, and therefore it's a very hot field of research, and it's very important that the less studied plant cannabinoids modulate the activity of these channels. And it's also important to understand how also the endocannabinoids, as I mentioned before, can activate these TRP channels. Anandamide can activate TRPV1 and antagonize TRPM8, the same for 2-HG. And very high concentrations of anandamide can also activate TRPA1. And it's interesting how the action of plant cannabinoids on these channels can be of therapeutic value. Recently, we've discovered in my group how these channels can be present in myoblasts. Myoblasts are indifferentiated muscle cells, and you can induce the differentiation. As you can see here, these are isolated myoblasts. They line up to form myotubes, which are really the component of the muscle fibers. And you can see how CBD and cannabidivarin can stimulate the lining up of myoblasts into myotubes. And they can also stimulate the expression of skeletal muscle differentiation. You can see how this effect is stronger with short time of incubation of myoblasts with the phytocannabinoids. If you incubate for too long, the effect goes down. This is usually indicative of a desensitization, meaning that in this case, even though, as I said before, sometimes it's difficult to understand whether it is the activation or the desensitization of this channel that produces a certain physiological effect. In this case, it was possible to hypothesize that is the activation that produces the differentiation of myoblasts into myotubes, because if you incubate for too long, this effect goes away, indicating that maybe when the channel is desensitized, it's not able to produce this effect anymore. And you see here how This can be important in therapy because if we take myoblasts from children with Duchenne muscular dystrophy, again, CBD and CBDV can produce the increase in these markers of differentiation. This is significant, statistically significant with both plant cannabinoids. And indeed, the TRP channels can also participate not only in the differentiation of myoblasts into myotubes, but also in the formation of myoblasts from satellite cells. Here you see how, starting from satellite cells, CBD can produce myotubes. How this effect can be blocked by a TRPV1 channel in mouse myoblasts. If we do the same and here you see the nice pro-differentiating actions of CBD and CBDV, and to some extent also of tetrahydrocannabivarin. And in the case of human cells, this effect is not mediated by TRPV1, but is mediated by another TRP channel, TRPA1. So TRP channels are important in the pharmacology of plant cannabinoids, and they can be important also in their use against some congenital disorders, genetic disorders such as Duchenne muscular dystrophy. Also, the well-known actions of cannabidiol as Epidiolex. As you know, Epidiolex is a drug that contains mostly 98% botanical CBD, and it's been marketed in the world now as an anti-epileptic agent against congenital disorders that are accompanied by seizures, and also it's been tested against autism. You see how in this particular model of seizures, which is the mouse maximal electroshock threshold model of generalized seizures, the CBD can induce a reduction of these seizures, and how this effect of CBD does not really happen in TRPV1 knockout mice, suggesting that CBD as Epidiolex can produce some of these anti-epileptic, anticonvulsant effects by activation of TRPV1. And there are many, many other examples through which non-psychotropic cannabinoids, in particular CBD, can produce therapeutically useful effects via TRP channels. Another important target for plant cannabinoids are the PPARs. The PPARs, as you know, are involved in the control of metabolism. PPAR gamma is involved in adipocyte differentiation, and here we could show that in fact these plant cannabinoids, cannabigerol acid, and cannabidiol acid, following an in vitro modeling study, we hypothesized that they could be a dual agonist for both PPAR alpha and PPAR gamma. There is an interest for this dual agonist because they seem to have less side effects than single PPAR alpha or PPAR gamma agonists in metabolic disorders such as insulin resistance. You see how these compounds can produce adipocyte differentiation, and this effect is mediated by PPAR alpha, PPAR gamma, which these compounds can stimulate in vitro, also in cells transfected with PPAR alpha and PPAR gamma in a luciferase transactivation assay. So PPAR, peroxisome proliferator activated receptors alpha and gamma. There are agonists for these compounds that are already on the market, have been on the market to treat steatosis and insulin resistance, respectively, can be targets for plant cannabinoids. So this is important because the fact that some compounds can produce actions by acting at different receptors rather than being magic bullet. So rather than being potent and selective for only one target, sometimes there is a therapeutic usefulness to have compounds that are polypharmacological. So they have less activity at different targets. And this is important because most of the pathological conditions that we want to treat and are still in need of treatment are deriving from the malfunctioning not of just one target, but of several targets. If we can have in the same molecule the capability, even mildly modulating of even mildly modulating more than one targets that are involved in disease, we could probably get to a more efficacious drug. Or if we know that some compounds can have effects on a particular target that can cause side effects, we can devise molecules that instead of activating that target, they antagonize the target and therefore abolish the side effects of other treatments. So it's important to investigate polypharmacology. And in fact, if you think of it, most of people with disorders take a combination of drugs in many cases to treat the same disorders. Hypertension is one example, but there are many other examples. So if we go back to the cannabinoids, we can devise for the cannabinoids a fingerprint. So we can put in a two-dimensional way on the y-axis their activation or activating or inhibitory action at different targets. Here you see the so-called pharmacological fingerprint of cannabidiol. Again, this is not really up to date because many other targets have been discovered for cannabidiol. You see that cannabidiol has the capability of inhibiting CB1 and CB2, but also activating PPAR gamma, TRPV1, TRPV2, inhibiting serotonin or activating other serotonin receptors, more receptors, glycine receptors. So we can design a pharmacological fingerprint for these compounds. And we can actually depict it in a way where we first try to understand, based on data found in the literature, what are the targets that need to be activated or inhibited in a given disorder, so-called etiopathological fingerprint. So, for example, assume that in different tissues, a given pathological conditions requires the activation of this target. So it's green to activate this target, it's red to repress it, it causes problems, but the other target instead needs to be repressed and doesn't need to be activated, or this other target mostly needs not to be touched. You can do this based on data on the literature on different molecular targets, depict in different tissues what are the requirements for several targets to be activated or inhibited in order to produce a therapeutic effect without producing a side unwanted effect. And then you can try to fit the what I showed you before, the pharmacological fingerprint of a particular compound, for example, a cannabinoid, at these same targets, okay? So you can depict, and this pharmacological fingerprint can be different in different tissues because, as you know, the environment at a given target, at a given receptor, for example, can change the sensitivity of this target to a given compound. So this requirement, this capability of a given compound to activate or inhibit certain targets can change in different tissues. And if you do this in the same tissues where you have done the etiopathological fingerprint, you can devise a plane like this and try to see whether it fits with the etiopathological fingerprint. If the points of this plane fall into the green areas of the etiopathological fingerprint, then you will predict to have a very efficacious and safe drug. So we've done that for multiple sclerosis because, as you know, cannabinoids are already marketed as nabiximols, as Sativex, against spasticity in multiple sclerosis. So the first botanical drug made out of the cannabis plant was this nabiximols, which contain more or less a similar amount of THC and cannabidiol. One spray of Sativex delivers 2.7 milligram and 2.5 milligram of THC and cannabidiol respectively, together with other botanical components of cannabis. And this has been standardized in a way that always the same amount from always the same type of plants is delivered, and it has been approved in several countries now against spasticity for multiple sclerosis. And the reason why this was devised, I mentioned it before, is because it was there was anecdotal data showing that those varieties of cannabis with high concentrations of cannabidiol would produce less side effects due to THC than those varieties like those used to make marijuana, where THC is the predominant cannabinoid. And this is also true for some of the psychotic experiences associated with cannabis smoking. So cannabis with high cannabidiol content is associated with fewer side effects, and yet it could still produce the same beneficial actions on disorders. So this is true at least for multiple sclerosis, for nabiximols, and we try to apply the model of the therapeutic handshake to Sativex. So again, we identified a series of receptors in different brain areas that in multiple sclerosis either do not need to be touched or need to be activated, like the GABA receptor, or need to be inhibited, like the Pha and MAG lipase, and these in different areas that normally control spasticity in the brain. So we could devise etiopathological fingerprint, three-dimensional fingerprint in different tissues for spasticity, and we fit the pharmacological fingerprint of the combination of THC and CBD in these different areas. And you can see here that in most cases, the plane deriving from the pharmacological fingerprint of the combination of THC and CBD in equal amounts fits with most of either the blue or the green areas. There are some exceptions, of course, which could probably determine side effects, but this basically predicts, and it was already known, that Sativex is quite efficacious against spasticity. And you could do the same thing with epilepsy. You could do the same thing with any kind of disorder. Here you see a series of targets that have been described that need to be activated or need to be inhibited in the hippocampus of animal models of seizures and epilepsy. And here you can devise the pharmacological fingerprint of CBD alone or in combination with two different types of epileptic anti-epileptic drugs like Keppra or valproate. So you get a pharmacological fingerprint of the combination of CBD and Keppra and combination of CBD and valproate that fits very well with most of these targets, with the requirement of most of the targets that from a literature search have been described to be important to determine epilepsy or to counteract epilepsy. So again, the therapeutic handshake model predicts that cannabidiol would be efficacious in epilepsy. And you can do that with other disorders for which CBD, for example, in this case, has not been tested yet in clinical trials, or ulcerative colitis. These are the targets. You cannot see them, but there are many targets involved in inflammation of the colon that either need to be activated or need to be repressed. You can, in this case, make a pharmacological fingerprint from the combination of CBD and 5-SA, which is mesalazine, a drug typically used for ulcerative colitis. And if you make the combination, you see that this combination fits very nicely with the etiopathological fingerprint of colitis. And this predicts efficacy for cannabidiol and 5-SA combination. We tested that combination in a model of colitis, and you see that doses of cannabidiol and mesalazine that are inactive in this model, when given together, would produce a very strong anti-inflammatory action in this model. So you can do that, and you can then test what you find in different models, as we've done here. And we always found that cannabidiol-enriched cannabis extracts are always more efficacious than pure botanical CBD against intestinal inflammation. You can do the same thing for pain. Now, this becomes complicated. Cancer pain. These are all the targets in different brain and peripheral areas that are involved in cancer pain. You can do the polypharmacological fingerprint for Sativex in this case, and try to fit it with these etiopathological fingerprints, and you come up with a response that is satisfactory but not so strongly satisfactory, predicting that Sativex could be efficacious against cancer pain. There have been two clinical studies with Sativex in cancer pain, and they did not produce completely satisfactory efficacy in the endpoint. So try to turn out to the end of my presentation. by telling you what's new on the study of the pharmacology of cannabinoids. You may have heard that we have a gut microbiome. Of course, you have the intestinal flora. So we and others have started to investigate whether plant cannabinoids or the endocannabinoid mediators communicate with the gut microbiome. Why? Because the gut microbiome is involved in many, many disorders, either by causing some disorders, but more often the gut microbiome is perturbed during pathological conditions. And this perturbation of the gut microbiome, the composition in each of the microorganisms that populate our gut, it's known as gut dysbiosis, can then feed back positively on the disorders, thus contributing, thus creating a vicious circle in some disorders. The gut microbiome is now being recognized as a major player in metabolic disorders, and I've showed you how the endocannabinoidome is important in metabolic disorders and in neurological disorders, in immune disorders, in inflammation. It is a fact that phytocannabinoids can be antimicrobial. Of course, they can modulate the activity of bacteria. This was shown by a group in Israel. Very interesting data. Not even very high concentrations of cannabinoids can produce all these series of actions in vitro against different types of bacteria, including bacteria that normally live in our gut. And there is now evidence also that in vivo, the plant cannabinoids, THC or THC alone or in combination with cannabidiol, or even only cannabidiol, as in this study from our group, can produce some of their beneficial effects, in part because they change the biology, the composition of the gut microbiota. And in particular, in the case of colitis, we showed that cannabidiol in combination with the fish oil can produce beneficial actions on the gut microbiota by increasing the relative abundance of those gut microbiota species that play an anti-inflammatory action and decreasing those that can contribute to inflammation, for example, by producing lipopolysaccharide species that are pro-inflammatory during gut dysbiosis. Even in obesity, there has been work by Kit Sharkey and by the group of Prakash Nagarkati showing that if you block CB1 receptors, either through a chronic use of THC, which causes desensitization, or with rimonabant, you, as I told you at the beginning, can produce, it was already known that you can produce anti-obesity action, but they showed that these anti-obesity actions of blocking CB1 receptors are partly due to effects on gut dysbiosis, on gut microbiota that contribute to the consequences of obesity, in particular at the level of the inflammation and of insulin sensitivity. So we basically studying the endocannabinoidome. I want to show you again this concept towards the end of my presentation. Just I want to go back to what is the endocannabinoidome. The endocannabinoidome is a series of endocannabinoid-like molecules. It includes the endocannabinoid system. So you see how anandamide and 2-AG are involved in the endocannabinoidome, but there is many endocannabinoid-like molecules like oledyl ethanolamide and other N-acyl ethanolamines or monoacyl glycerols, N-acyl amino acids, N-acyl neurotransmitters, primary amides, which can either activate or inhibit the targets of the endocannabinoidome. And the endocannabinoidome, as showed you, explains the pharmacology of all plant cannabinoids, not just of THC. The endocannabinoid system is good to explain the pharmacology of THC. The endocannabinoidome is much better to explain the pharmacology of other plant cannabinoids that are being now tested in the clinic. But more interestingly, going back to the microbiome, some receptors of the endocannabinoidome, not so far, not yet CB1 and CB2, but other receptors such as GPR119 or GPR32, which, as you can see, can be modulated by other endocannabinoid-like molecules, are also activated by molecules produced by gut microbiota. You see here how these molecules can be very similar to some of the N-acyl glycines or endocannabinoid-like molecules that are produced from the mammalian host. And by producing these molecules, the gut microbiota interact with the same receptors as the host endocannabinoidome to produce either beneficial or negative effects. And this is important, and this is another important concept I want to pass today. This is important for at least metabolic, inflammatory, and brain disorders. And it's important to understand how the receptor members of the endocannabinoidome are not all playing the same role. For example, this is true also for CB1 and CB2. We know that CB1 has a pro-inflammatory role in some cases. It induces food intake, reduces energy expenditure. It basically contributes to obesity and metabolic disorders. Many of the other receptors of the endocannabinoidome do exactly the opposite, including CB2. One thing that CB1 does is to increase the intestinal barrier permeability, and therefore, when you have a gut dysbiosis, an imbalance of the gut microbiota, some of the pro-inflammatory molecules produced by the gut microbiota can pass through the intestinal barrier and produce negative effects. CB1 promotes these negative effects. Many of these other receptors, including TRPV1 and GPR55, GPR119, which are receptors for the endocannabinoidome, do the opposite action. And this is important not only for metabolism, but also for neurological and neuropsychiatric effects, where instead it is more frequent that TRPV1 channels play a negative role and CB1 and CB2 receptors play a positive role. It's important to investigate the actions of all the receptors of the endocannabinoidome, not just CB1 and CB2, if we want to develop drugs from either plant cannabinoids or from inhibitors of the degradation of endocannabinoids. And maybe one of the reasons why inhibitors of the degradation of endocannabinoids have not been successful so far is because when you inhibit the degradation of anandamide, you also inhibit the degradation of other N-acetylphenylamides, which have different targets, and therefore you can create conflicts in the indirect pharmacological actions of the inhibition of the degradation. This could explain why in mice you have some beneficial actions that you don't see in humans. So these are my take-home messages. I think I'm almost on time. Just to remind you that cannabis is not just marijuana or hashish or skunk. There are several varieties of cannabis that differ from each other for their composition in phytocannabinoids and THC content. So far, until the end of last century, we focused on THC. In fact, in Brazil, there have been several studies also on cannabidiol before this century. So Brazil has been a precursor in the pharmacology, in the study of the pharmacology of non-THC plant cannabinoids. There is more than 100 these compounds, THC being the only euphoric psychotropic component, and the only phytocannabinoid that potently activates CB1 and CB2 receptors. So the endocannabinoid system, CB1 and CB2, is not enough to explain the pharmacology of all cannabinoids. And we are interested in the pharmacology of all cannabinoids because through their multi-target and multimodal action, these phytochemicals seem to be more promising from a therapeutical and clinical point of view than pure THC. However, we need models as the one I showed you in this presentation of the even more sophisticated models than the therapeutic handshake to try to predict not only their efficacy but also their safety, exactly because they have several targets. And I showed you that the phytocannabinoidome interacts with the expanded endocannabinoid system rather than the simple endocannabinoid system, and we refer to this expanded endocannabinoid system as the endocannabinoidome. However, it is also true that the phytocannabinoids that are different from THC sometimes interact with other receptors that are not part of the endocannabinoidome because they are not modulated by endocannabinoid-like molecules. And a typical example is the serotonin receptors or the glycine receptors that are modulated by plant cannabinoids, but not necessarily by endocannabinoids and endocannabinoid-like molecules. And beyond side effects and epidiolecs, several other potential clinical applications can be forecast for CBD and other non-THC cannabinoids in chronic pain, skeletal muscle dystrophies, skin and bowel inflammatory disorders, and even cancer. I didn't have time to talk about cancer, but definitely cancer is a very promising target for non-psychotropic cannabinoids. And this range of indications may even be expanded further to other disorders in which gut microbiota dysbiosis participates. And it's important that we investigate the actions of phytocannabinoids. These drugs are given orally, and anything that is given orally before encountering our cells, they will encounter the gut microbiota, and they can change the gut microbiota, they can be changed by the gut microbiota. So we really need to investigate the interactions of cannabinoids and endocannabinoids and endocannabinoid-like molecules with the gut microbiota. I want to thank the groups. I have the luck and the privilege to lead in Quebec City and in Pozzuoli. In Italy, slightly different climatic conditions, but they're both beautiful places to work with. And thank you very much for your attention. Please tell me that you were listening.
Yeah,
we didn't do any double check whether you could hear my voice.
No, everything worked just fine.
Oh,
We would like to thank you very much. There are actually 2 rooms full of people watching the talk here at the Institute. And right now, Lucy is collecting the questions, and I'll project them so we can
okay,
have a little bit of discussion.
I'll go back to the, okay. I'm back to the...
Yeah, it's working just fine.
Okay.
We just have to wait for the questions. And everyone here on the chat, we're live on YouTube also, and there are people all over watching us.
Oh my God.
Yes, big pressure. So people commented on the video, and they said the talk was great and thanked us very much. Thanked you very much for
it's been my pleasure.
Talk. Yeah, we hope to have other encounters here. I'm just waiting for the questions because they're in the other room, and then I'll project them here.
Yeah, I hope some concepts were a bit complicated, but I think I managed to convey the idea that this is a very exciting field of
Yes.
work, but also very challenging to understand. But definitely, we need to do work on the other cannabinoids as well.
We have a question here from our professor from the Institute. Mm-hmm. What is the strongest predictor of cannabis-inducing psychosis in teenagers?
Yeah, as you know, there has been for a long time this misconceived idea that it's enough to smoke cannabis with high concentration of THC, meaning with the capability of strongly activating CB1 receptors. Now, THC is not as strong as some synthetic compounds that are present in spice. Those are very dangerous. THC is a partial agonist for CB1 receptors. But nevertheless, it can produce serious side effects, not only at the level of the brain, but also at the level of cardiovascular actions, particularly. But really, now we know that not everybody is susceptible to the psychotic events triggered by marijuana smoking and THC. There have been several genetic studies, and definitely one of the genetic variants, SNPs, single nucleotide polymorphisms, that have been associated with the genetic susceptibility to psychosis following marijuana smoking is the one of COMT, catechol O-methyl transferase, which is an enzyme involved in dopamine degradation. So it's interesting how those people who have this genetic polymorphism in COMT are more susceptible. Probably it's because, as you know, psychosis and schizophrenia are strongly connected with dopamine, and this could be the link. But definitely, as far as I know, I'm not really up to date to the knowledge in this. The presence of this variant is one of the strongest predictor of cannabis-induced psychosis in teenagers.
Thank you very much. We have another question. Professor Ricardo Reis is asking about personalized medicine. Is it possible to use therapeutic handshake regarding to rimonabant to ameliorate metabolic effects and diminished psychiatric effects?
Yeah, I actually see the I can see the questions, Beatrice.
Yeah.
So don't worry repeating them.
All
Uh,
right.
we can save time. Yes, this is a very interesting question. It's the therapeutic handshake is a bit difficult to use for personalized medicine because it's done by using data in the literature. So we would need a way to quickly collect all that information from different patients. Definitely can help us understand why some patients are more or less responsive to cannabinoids. Rimonabant as well, so CB1 antagonists. But what we're trying to do now, more in terms of personalized medicine, is to first understand if some of the actions of cannabinoids are mediated by the gut microbiome. And if they are, since it's much, much, much easier to profile the gut microbiome, we can use the information from the gut microbiome to try to devise different drugs. If we can demonstrate that some of the effects of cannabinoids or rimonabant are strengthened and or reduced by a certain type of microbiome, of gut microbiota composition, we can easily assess that in patients before we start treating them, and we can do more personalized medicine. I know that many people are doing that, also using the metagenomic studies from the host and not just from the microbiome. But we think that can also be amenable to personalized medicine because we can also simulate the human microbiome ex vivo, which cannot be done with the human, cannot always be done with the human tissue or the human biopsy, because we can put fecal samples from patients in a machine that simulates different sections of the gut, and we can actually study the effect of drugs or effect of nutrients on the microbiome, and we can see whether donor can have different response from another and predict whether this would lead to better responses or to side effects.
Thank you very much. We have the last question. It's from Professor Isis.
What is the impact of milk cannabinoid on gut microbiome and development? Okay, milk cannabinoid, I guess it's the endocannabinoids or the milk of women who are taking cannabis. In any case, we know, because we know that milk is a very rich source of endocannabinoids, particularly 2-HG. And we're actually investigating now, this is a very interesting question, we're investigating whether there is, first of all, if the mother can affect through the milk endocannabinoidome the gut microbiome of the child, and whether this can explain some pathological conditions that you have during lactation. And we guess that the consumption, since we've seen that many people are seeing that plant cannabinoids can also change the gut microbiome, there could be an interaction between the endocannabinoid or the cannabinoid taken from smoking cannabis and the milk microbiome in development. It's a very difficult question to answer because the models, the animal models to investigate this question would require treating the mothers, the dams of mice, and then use the pups during prenatal or postnatal development to see whether there is an effect. But sometimes the ethical committees do not allow to do that. But this is definitely something that is very important to understand. Because I tell you why, because we all worry about mothers smoking marijuana. This is something that of course should not be done, but we worry about the cannabinoids, but you can affect the milk endocannabinoids by eating the wrong fatty acids. So if you eat a lot of stearic acid or palmitic acid and you forget to have docosahexaenoic acid, omega-3s, omega-6, you can change the endocannabinoidome in the milk, and you can then change the child development through the milk.
We have to finish with the questions now because of the time, but we would like to thank you very much for the talk. I'm sure everyone enjoyed it very much, and we hope to have an encounter in person in the future here at the Biophysics Institute, and we are very grateful for the talk.
Thank you very much, and thank you again for the invitation. And as I said, you can contact me by email if you have any further questions.
All right. Thank you so much, Professor.
Thank you. Have a nice day. Bye-bye.
Bye.
Hope to see you soon. Bye-bye.
Yeah, we too. Bye.